So I'm going to talk to you today about dopamine and the value of work. And I first of all want to acknowledge some of the people in my laboratory and beyond who helped with this work, especially two very talented graduate students, Arif Hamid and Jeff Pettibone. So my laboratory studies primarily neural circuitry of the basal ganglia. And I won't in this talk need to go through in great detail what the basal ganglia all their connections and components. The basal ganglia are a central aspect of all vertebrate brains, and yet we understand very little about what they do. If you forced many basal ganglia experts, so-called experts, into a room, and you locked the door and you said, you're not allowed out until you agree what they do, they might come up with something like this, that the basal ganglia are important in deciding what is worth doing, some role in selecting and energizing actions based upon what's worked well for you in the past, learning from a history of reinforcement. And the subject of today's talk is one of the main components of basal ganglia function, which is called dopamine. And most people are aware to some degree of dopamine. They've heard of it one way or another, and some of the ways that people have heard of it is because if you lose this dopamine projection from the midbrain to the striatum, then that causes the key symptoms of Parkinson's disease. And conversely, drugs that enhance dopamine release within the striatum tend to be abused drugs, cocaine, and so forth. And so dopamine comes into the striatum from these enormous axonal projections originating in the midbrain. And each dopamine cell in the midbrain provides input to maybe 100,000 striatal cells. So it's supposed to be a fairly divergent broadcast signal. And what signal do they send? So everyone knows or thinks they know that dopamine has something to do with reward. And this has been studied for many decades now. But it doesn't just signal reward in some simple way. So let me show you, first of all, a seminal set of experiments from Wolfram Schultz and his group, where they were studying, first of all, the response of a dopamine cell to a reward that is unexpected. So if you have a monkey and you squirt fruit juice into its mouth unexpectedly, what you see is an increase in the firing rate of a dopamine cell. And that's both shown on the right side of the picture here. And if you have a cue that's familiar, reasonably familiar, but isn't predictive of reward, then dopamine cells tend not to care about it. But if you pair these things together, so the cue reliably predicts the reward, what you see is that after the monkey has learned this association, after he's been presented with this pairing of events many thousands of times, what you see is that the dopamine cell starts to respond to the cue and actually stops responding to the reward itself. Even though the fruit juice is still rewarding, the monkey still likes the fruit juice and still licks the fruit juice avidly. So dopamine is clearly doing something more than just signaling you just got a reward. Okay? So for dopamine cells to respond, there has to be something surprising, at least in this framework. And this is just showing the same kind of data in a different way. Each little tick mark here is one action potential from a dopamine cell. And this is just showing that if you provide an expected reward, then you don't see too much of a response. If you provide it at a delay, what you see is, first of all, a sort of pause in firing and then an extra firing after it actually happens. So dopamine cells are sensitive not just to getting a reward, but to the timing of that reward as well. And the sort of dominant paradigm in the field of dopamine research is to think of the dopamine signal as conveying what's called a reward prediction error, RPE. And that means that it's signaling whether things are better or worse than you thought they were going to be at that particular time. So it's an important concept in current neuroscience. So if you imagine, if you want to calculate a reward prediction error, you need, first of all, to have a reward prediction. And in turn, you can use the error in that prediction to update that prediction. So you can imagine a cycle where you make predictions, you find out the consequences, what actually happens, and you use those events unexpected events to update your predictions. And that's one important aspect of learning. And one reason why this way of thinking about things has caused a lot of excitement over the last decade or two has been this sort of very fruitful dialogue between neuroscience and computer science. So computer scientists, starting a couple decades ago, started taking some older ideas from the psychology literature and developed a set of machine learning algorithms, which are called reinforcement learning. And they're actually very useful and actually being used in all kinds of actual computer systems and robotics these days. And so that's been extremely, the fact that you can actually use these algorithms to achieve useful things has been a sort of validation of these ideas and has led to really driven people to want to understand them better in the brain as well. So I'm not going to give a whole lecture on computer science of reinforcement learning today. I'm just going to try to present some basic concepts so that you'll need for the rest of the talk. And it shouldn't be too bad. So just imagine you have an agent acting in the world, and the agent can be in one of several different states. So you might say, well, when I'm faced with this set of cues and this context, that defines one state that I'm in. And each of these states has some value, and that value is how much reward do I expect to get in the future given that I'm in this state. Okay. And when you change from one state to another, if you change to a state that has higher value and you weren't expecting that to happen, then you have a reward prediction error. So for example, you're sitting around and then you get a cue that says reward's coming. You say, oh, okay, now I'm in a state that reward is coming soon. And that means that I just got a reward prediction error. Things are better than I thought they were going to be the moment beforehand. All right, and again, as I said before, you use the reward prediction errors to update the values, either of states or actions you might take in a certain state. All right, I hope that's clear. So lots of people have had lots of ideas about how to map these concepts onto certain brain systems and structures. This is, I'm going to show a couple slides from Kenji Doya, whose stuff I love, not because it's right, but because it's simple and elegant and is useful heuristic. So one way of thinking about brain organization, or a good chunk of it anyway, is to imagine that the cortex is representing certain states. That feeds information about those states into the striatum that evaluates those states. So it has learned the value of being in each of these states. And the dopamine cells provide an input that helps to update those weightings, update the values assigned to each state. All right. But so despite the sort of prevalence and predominance and impact of reward prediction error ideas of dopamine, not everyone agrees that that captures the key functions of dopamine. So one person who doesn't is my esteemed colleague here at Michigan, Ken Berich, who has argued for a long time against learning ideas of dopamine and instead saying it's missing something critical about dopamine's role in motivation. So just as we've known for a long time, there's something about dopamine and rewards. We've known there's some relationship between dopamine and motivation. If I were to break it, this is a webinar, so I can't just hand each of you some amphetamines, but if I were to do so, the obvious thing that would happen is not so much that you were learning better, but that you might be more excited and have more greater psychomotor arousal. So something's going on there that's not simply captured by the learning framework of dopamine. And many different terms have been used to describe this possible role of dopamine and motivation, something to do with approach behaviors, incentive salience, wanting things. We're going to address some of those over the next few minutes. So one way of thinking about this debate is to imagine, well, what is striatal dopamine good for? Again, if you have some hypothetical agent in some environment, the agent performs actions and they affect the environment, and then the agent gets feedback from the environment about whether the actions worked out well or not. So is dopamine fundamentally about this feedback process? Dopamine fundamentally about this learning, about the consequences of your actions? Or is dopamine fundamentally about the performance aspects of things? Is it driving you forward to perform certain actions and act in certain ways? Or both? And if it's both, how is it both? Why are these? How can one signal do these two different things without whatever's listening to the signal getting confused? Okay. So one way that people have tried to reconcile these ideas is to imagine that dopamine is doing both learning and motivation, but on different timescales. So this is one influential paper proposing that dopamine's role in motivation was really happening on slower timescales, the so-called tonic dopamine. So in this framework, phasic, that is brief fluctuations in dopamine, are used as learning signals, and more prolonged changes are used as a motivational signal. Specifically, perhaps this tonic dopamine signals the rate of reward. That is, how frequently are you getting rewarded for a given course of action? And this would be a useful signal to have, because it might tell you whether engaging in that course of action is actually worth your time and effort. So we set out to test some of these ideas in various ways. And this is a sort of typical experimental setup in my laboratory. This particular rat is wired up for single unit neurophysiology, which I'm not going to talk about today, but it's the same operant chamber. So we have a rat in a box, and we have a set of holes on one side, and one of those holes can light up, and the rat has to go stick his nose in the hole, something like this. Okay, so this would be the start of the trial, and the hole lights up. That's going to be an important event. And then he goes and sticks his nose in that hole, and the time it takes him between the light coming on and him sticking his nose in the hole, we're going to call the latency. And for reasons I'll show you in a second, we're going to use that as an index of how motivated the rat is to play this game. All right, so the rat sticks his nose in the hole, he waits for maybe somewhere between half a second and a second and a half, and then he gets a burst of white noise that are used as a go cue, and at that point he has to choose, and he chooses either to go left or right. Okay, so he pulls out and sticks his nose in one of the side ports. In this case, he chose right. And on some trials, just as he enters that side port, then a sugar pellet is delivered behind him in the food hopper, and he can go and collect that sugar pellet. But he doesn't get rewarded at every trial, so it's a stochastic task. Sometimes he gets rewarded, sometimes he doesn't. And the left and right choices don't always have the same probability of being rewarded. So they change. So he has to adapt to those changes. So here's an example. This would be the beginning of a session, and purple tick marks indicate when the rat made a left choice, and green tick marks indicate right choices. Tall tick marks are rewarded, and short tick marks are unrewarded. So in this case, he tried going left, got a reward. Tried left again, no reward. Left again, no reward. Try right, no reward, and so forth. So this would just be the beginning of a typical session. A full session might look like this, several hundred trials. And you've got the reward probabilities listed at the top, starting at 50-50 and then shifting 10-50, 90-50, and so forth. And what I hope you can see from the running averages shown below is that the animal adapts his left or right choices to whichever side is currently producing the highest likelihood of reward. Okay. But he's not just adapting his choice behavior, he's also adapting his vigor, his motivation to play this game. And you can see that here. So now I've just shown the same data, but just ignoring whether it was left or right choices, and showing just so you can just see more clearly the density of tall tick marks, which is the rewarded trials. And I don't have a pointer here, but if you look at about 130, 140, you can see there's a period of time when he's getting lots of rewards. Almost all the trials are being rewarded. And so the black line beneath is showing the sort of density of rewards. And the blue line, which is on an inverted scale, is showing his latency. So I hope you'll be able to see, if you look at this for a few seconds, that when he's getting a higher density of rewards, his latency is shorter, meaning his motivation to engage in this task is higher. So this is just one example session. We can look at it over many sessions. So this would be many sessions for multiple rats aggregated together. And what you can see is that once at the beginning of a block, he doesn't necessarily know which way to go. But then as the block goes on and he has more experience of the current contingencies, then he shifts his choice to being towards whichever one is better at that time. So that's the left-right choice aspect of things. And then here's the sort of motivation aspect of things. So if he's received many, many rewards, so 10 out of the last 10 trials have been rewarded, then he's very fast to engage in the next trial. Whereas if he's not received any rewards in the last 10 trials, he really can't be bothered to play this game. And why should he? So the point here is there's two different aspects of adaptive behavior within the same task. And just to elaborate one more thing, so why is it that having a higher reward history produces shorter latencies. How should we start to break that down in terms of psychological decision processes? It's the same data as before. So you can show how likely he is to initiate the trial at any given moment after the light comes on as a function of reward history. So this middle plot is basically a survivor curve, the fraction of trials as a function of time for which he has not yet initiated the trial. And then you can also show that on the right is a hazard function. And so basically the idea here is that the shorter latencies arise from an earlier decisions to initiate an approach to begin the trial. Right? And the reason I'm bothering to show this is because it'll become important later on in interpreting some of the data. Okay. So now, having established this behavioral task where we have adaptive choice and adaptive motivation, we can study how dopamine relates to different aspects of the ongoing decision making. We can say, is it true that tonic dopamine correlates with reward rate? And is it true that phasic dopamine signals reward prediction errors in this task? So we're going to use two different methods for measuring dopamine. Measuring dopamine in general is a big pain in the neck. Sometimes some methods are more of a pain in the neck than others. We're fortunate here in Michigan to have some leading experts in some of these different ways of assessing dopamine. So one of those experts is Bob Kennedy. His lab is a leading lab in microdialysis. So this is basically collecting tiny drops of brain juice from the striatum, ventral striatum, every minute. So 1 minute is much faster than conventional microdialysis. It's still not super fast. But one advantage of the microdialysis procedure, as Bob Kennedy's lab does it, is you can detect not just dopamine, but many, many other chemicals at the same time. Right? We're going to use this microdialysis technique to assess what we call tonic dopamine. And then we also have another technique, and this is one of the people here who's a leader in this field is Brandon Aragona, in his lab has expertise in fast-scan cyclic voltammetry. So voltammetry is the technique that allows you to measure dopamine 10 times every second. It does have, however, some disadvantages, which is you can't measure 10 times every second for many, many minutes, and have a stable baseline. So signals tend to drift over a while. So you're really looking at fast fluctuations. So we have two complementary methods. One is better for looking at tonic dopamine. One is better for looking at phasic dopamine. So here's just an example of what a microdialysis sample analysis would look like. So here's the measured chemicals from one single drop of brain juice, and we're measuring, as you can see from the top, a whole range of things: dopamine, the dopamine metabolite, 3-MT, norepinephrine, and so forth, serotonin. Basically all the major low molecular weight striatal neurotransmitters, and many of their metabolites as well. And so we can ask a simple question: does the time-varying signal, minute-by-minute levels of each of these neurochemicals co-vary with various behavioral things that are going on? And again, we're trying to test, first of all, this hypothesis that dopamine co-varies with density of rewards, reward rate. And so when we do that, we find that, yes, we have a highly significant relationship between dopamine and reward rate, a slightly less significant relationship between the dopamine metabolite and reward rate, and no other brain chemicals correlate with reward rate. So this seems good so far, but we need to also make sure it isn't just a spurious correlation based upon some other relationship that's going on. So we could say, for example, does dopamine just correlate with psychomotor activation? Like if you're just moving around more, if you're making more attempts, does that correlate with dopamine? And it does a little bit, but not so well, and that little bit is entirely accounted for by the reward rate correlation. Dopamine has also been proposed to correlate or drive exploitation versus exploration of alternative options. We don't find that to be true. I'm not going to go into detail of that now. And you might also want to know if dopamine just correlates with the total number of rewards you get. And it turns out there are various things in the striatum that do care about how many rewards you've gotten in this session, but dopamine doesn't care at all. Okay, so far that seems good. Dopamine is specifically linked with reward rate, and it seems to support this idea that there's a dopaminergic reward rate signal that's used to decide how hard to work. But, is it really the case, just because we're measuring something that's on our measurements on a relatively slow timescale, doesn't mean that the dopamine signal in the brain is changing on a relatively slow timescale? So we wanted to know whether this slow dopamine signal is really different to the faster timescale fluctuations, or is it just like a time-smeared version of the same thing? So we turned to the voltammetry side of things. I think I'm not, in the interest of time, going to go into great detail into how the voltammetry technique works, when it does work. But nonetheless, these sort of puffs of purple that you see in the top plot here are sort of indications that you are detecting specifically dopamine release events, increases in dopamine. So here's a little video. I hope you can see this. There's a rat in the box, and one of the lights is going to come on. He sticks his nose in the hole. He waits. The side lights come on. He chose left, and he can now go and collect a pellet. And in the bottom scrolling trace, we can see the real-time changes in dopamine that are going on at the same time. All right, he chose right, did not get a reward. Okay, so you can see that things are fluctuating up and down, in some way that seems linked to behavioral events. But of course, it's hard to sort of do that correlation just in your head, and so we're going to analyze that properly now. Okay, so here we have all the trials from one session, one voltammetry session, and we're aligning the dopamine signals on various behavioral events. So this first event is just the light coming on to signal that the trial is available to the rat. And the second event here is just him actually inserting his head into the illuminated port. And I hope you can see that there's a slight increase in dopamine just before he puts his nose into the center port. And then for the next two panels, I'm going to break trials up into those in which he got the cue saying, yes, this is a rewarded trial, and those in which that cue was omitted, and so he knew he was not going to get a reward. And what happens when he finds out he's going to get a reward at side in, as you see this series of bumps upwards. The dopamine increases after that side in click, saying, yes, food's being delivered, and increases further as he runs over to collect the food pellet from the food port. Then once he actually grabs the sugar pellet, dopamine decreases pretty fast. Okay. So remember, one of the key things we're trying to assess with the voltammetry is, do fast fluctuations in dopamine really encode reward prediction errors? So here I'm just showing all the data combined from across multiple rats, and again broken up into rewarded and unrewarded trials. And if you further break up the data by how surprising the reward cue was, then you see this. So in all the red traces here, the same event happens. And in each case, he's getting the same cue that says, all right, now food is being delivered, you can go get it. But if he wasn't expecting it, because so very few of the last 10 trials have been rewarded, then it seems like you get a much bigger increase in dopamine. Okay. Conversely, if he finds out that he's not going to get a reward, but he has gotten lots of rewards recently, then you have a bigger negative reward prediction error, and dopamine seems to fall to a lower level. Okay. So far this seems very compatible with the idea that fast fluctuations in dopamine are signaling reward prediction errors, at least around this side-in event. However, the overall time course doesn't obviously map onto some reward prediction error account. I mean, what you're seeing is increases towards the beginning of the trial as he runs over to initiate the trial, and then further increases as he runs over to collect the food pellet. And having been told that the food pellet is available and then going to get it doesn't seem like anything particularly surprising is happening. So why should you have any reward prediction error there? And we're not alone in showing this kind of thing. I'm just going to briefly show some examples. A couple from Paul Phillips and Mitch Reutemann and their colleagues showing that with the same voltammetry technique, you see these increases in dopamine in conjunction when animals run over to initiate certain things. So, for example, running over to push a lever to get cocaine or to get food. All right? The increases in dopamine are happening before you actually complete that lever push. And then a couple of years ago, there was a paper from Anne Grabiel's lab showing increases in dopamine over several seconds as animals ran along a maze arm in order to collect an expected reward. Right? It's sort of ramping up in dopamine. So what's going on? What have we seen so far? So we see these phasic, timescales of seconds or less, changes in dopamine that accompany motivated approach behaviors. And this is happening at the beginning of the trial and approach to the food port. We also see these fast fluctuations in dopamine that seem to signal reward prediction errors at the time of getting feedback about future rewards. And then we have the so-called tonic dopamine that seems to co-vary with reward rate. So, is it really the case that these are all different aspects of dopamine and the brain somehow has to sort them all out? Or, can we bind these ideas together? Can we explain all these different things with one set of ideas? And obviously I wouldn't say that unless I thought that we could. And what I'm going to argue to you is that dopamine across multiple timescales is signaling the value of work, the value function. And so I'm going to have to spend a few minutes explaining what I mean by that. So a sort of fundamental concept in a lot of economics and decision-making literature is temporal discounting. And this is basically the idea that a reward right now is better than a reward sometime in the future. So imagine that you have to pass through a sequence of states in order to get to a reward. Right? So reward isn't just right there. You have to work for it. You have to go through states 1, 2, 3, 4, 5, 6, 7, 8, 9 before you can get the reward. You pass through these unrewarded states to get to the rewarded state. So what is the value of being in each of these states? You can imagine that it's more valuable to be at state 8 than it is to be at state 2. So you can imagine these discount curves. The value of being you increases as you get closer and closer and closer to reward. Okay. And these temporally discounted values just are the state value, the discounted reward that in the future you expect to get. So in some sense, sort of central to reinforcement learning algorithms is the idea that you use your experience to make estimates of these values and then to adjust these values. And then having got these values, you can use them to make decisions. For example, decisions about whether it's worth playing the game or not. So that's what I just said, that the time discounted value is a useful decision variable. So if, for example, reward is further away in time, or smaller, or more uncertain, then you should be less motivated to work. So imagine, for example, you were on this curve, this value function leading up to the 0.5 level, then you'd be less motivated, less likely to decide to work than if you were certain the reward was coming at the end. All right. But let's start to get things a little more similar to our actual experiment. So supposing you're running through a sequence of states, and in the middle of that sequence you get a cue that says, yes, this trial is a rewarded trial. What should the value look like in that sequence of events? Right? So you're passing initially along the curve that leads up to 0.5, and then you get a reward cue, and now the value of being you suddenly jumps up to the time discounted value of the full reward. And if you find out you're not going to get a reward, value should sink down low. Okay. So there's one more interesting thing here, which is that depending upon your expectations of rewards, the size, the amount you jump up is going to be different. So if you were very confident you were going to get a reward, say you were 75% expectation, and now you find out, yes, you are, you only have a small jump up, a small delta there. Whereas if you thought it was very unlikely you're going to get a reward, only 25% chance, now you have a big jump up. So the size of this jump is a reward prediction error. So the point is that if you can signal value in a way that dynamically changes from moment to moment, then the sudden changes in value also convey reward prediction errors, and that might be useful. Okay, so I'm again going to argue that the real dopamine signal is similar to a value function, but the real task that we did is more complicated than the sort of cartoon conceptual situation I just presented. In part, the reason for that is because the timing between events varies. So in order to assess this relationship between value and dopamine more precisely, we created a model that evolves in real time and looks something like this. So it's called a semi-Markov decision process. It just, again, consists of a sequence of states and with variable timing between different states. And each of these states has a value. That value is learned by how far away it is from the actual rewards. So we're really just running a reinforcement learning algorithm to both derive and update state values. So here's an example. So the top trace here, the black trace, is real dopamine signal obtained from the ventral striatum. And in red, the RC means when he got a cue saying this trial is rewarded. And blue is when that reward cue should have come but didn't, indicating that he's not going to get a reward for a while. And below this real dopamine trace, we've put in some of the model variables. So V is the state value. And what I hope you can see is that as you progress through a trial, you have an increasing value because you know that you're getting closer, and you think it's likely that you're getting closer and closer to rewards. And below that in magenta, we've put the reward prediction error as delta. And so if you get a reward cue, and for example, in the first trial that was very unexpected, then you have a big positive reward prediction error. And the size of that prediction error goes down as you get successive rewards. But then if you get a omission trial, then you see negative reward prediction errors. Okay. And so the average state value that you see in this model resembles pretty well the time course of dopamine. I hope that's pretty apparent. And okay. So let's see. So this is sort of one of our conclusions, and we'll certainly talk more about that in a minute. So what about the reward prediction errors? So I argued a minute ago that you could encode reward prediction errors not as absolute changes in dopamine, but as relative shifts in the value function. Okay. However, there's a sort of big difference in encoding reward prediction errors this way compared to the usual Schultzian model of the usual model of reward prediction errors. So here, reward prediction errors are shown as a jump up to a fixed level, the top of the red curve there, from a variable level. So that's very different to what I showed you at the beginning, which was sort of a steady firing rate of dopamine cells, and then a sudden jump up, depending upon how surprising the reward was. Now, I showed this picture before, arguing that dopamine seemed to do something like a reward prediction error, but the trouble is that I kind of told a bit of a fib. Well, not really a fib. To produce this image, you process these voltammetry signals exactly the way that everybody else does it, which is you define some baseline period before the trial, and you suppose that we're going to call that 0. And now we're looking at fluctuations away from that baseline period. And when you plot the data that way, it appears to show that more surprising rewards, that's the dark red here, evoke higher absolute levels of dopamine. But the same data, you could, instead of taking a baseline epoch before the trial, you could take one in the middle of the trial. It's equally compatible with the data. Right? So it's the same data shown relative to the dopamine levels later on. So presented this way, it could be compatible with our model idea that the reward prediction error is really a jump from a variable level to a fixed level. Well, if the data is equally compatible with both of these presentations, which one is true? How do we resolve that? And we decided to resolve that by looking at how dopamine changes between successive trials. All right? So a conventional reward prediction error model, imagine that you have 2 rewarded trials in a row. So first of all, you haven't been rewarded for a while, and now you get a rewarded trial. You should have a sizable positive prediction error. And if the next trial, the very next trial, is rewarded as well, then you should still have a positive reward prediction error, but it shouldn't be quite as big as the first one. And then again, it should sort of decay over time, because you're less surprised to get a reward. In the alternative account, the value-based account, your expectation is low at the beginning, you get a reward, but now your expectation of reward is higher, and that's seen as a higher baseline level. Once you actually find out that you are going to get a reward, it should go up to the same level and not higher. Okay, so these are the predictions that these different models make. Change in dopamine at different times as a function of reward history. And so when you do this, what we actually find is that the state value model is much closer to the truth. You can see in the bottom left, I hope, that the dopamine change is happening within a trial, and then a rewarded trial, that is. And then on the beginning of the next trial, you're starting from a slightly higher level. But you don't go up. The reward prediction error doesn't decline. Sorry, the level of dopamine that you reach doesn't decline in the second trial, as it should in a conventional RPE account. Okay, so I won't belabor this point, except to say that this overall increase in dopamine also explains why with the microdialysis, with the slower timescale data, we see a relationship to reward rates. If you have higher density of rewards, you have a higher expectation that the next trial will be rewarded, and that's reflected in a higher level of dopamine. Okay, so the argument to this point is that dynamic changes in dopamine can be used to do two different things. They can be used to influence motivation, telling you whether it's worthwhile engaging in some effortful activity, for example, or potentially as a learning signal, conveying reward prediction errors, or both. Right? So the argument is that dopamine is involved both in the reward predictions and in errors in those predictions. So this is showing that these signals could be used that way, but it isn't showing that they're actually used that way. All right? So we wanted to test whether that's really true. So the predictions here would be, if dopamine is involved in motivation, then if you alter dopamine, then you should have an immediate impact on the rat's behavior. Whereas if dopamine is involved purely in learning, then changing dopamine, especially at the time of reward feedback, should update values that affect behavior on the next trial, not really on this trial. All right, I hope that's clear. So we turn to an optogenetic method to assess these ideas, and in brief, we're using genetically modified rats together with channel rhodopsin viruses to allow us to have selective control of the firing of dopamine cells. And we use a blue laser to tickle the infected dopamine cells and look at the resulting dopamine release in the forebrain. So this is just an example of that. If we give a train, that blue train of pulses, just for half a second, to tickle dopamine cells, and then measure the resulting dopamine release, you see a nice punctate pulse of dopamine release. And we tune the laser parameters to try to get a pulse of dopamine release that is truly phasic, so it has about a similar duration and magnitude to what you'd see with an unexpected reward. Right? So we're going to give this pulse of extra dopamine, or block dopamine with a different technique, at different times within the trial. So we're either going to give extra dopamine at the beginning of the trial as the light turns on to assess the impact on the animal's motivation as measured by this latency, or we're going to give the laser at the time the animal puts his nose in the side port and finds out if he's going to get a reward or not. Right? And that's a time which we might update whether the left or the right choice is a good thing to do. And in any given session, we're only giving laser stimulation at one of these two times, and on a randomly selected 30% of trials. So, first of all, what happens if you give dopamine at the time of reward feedback? Well, what happens is that on the trial after stimulation, he's more likely to repeat the same choice. So if he goes right, and as he finds out whether that works or not, you give extra dopamine, then he's more likely to go right on the next trial. So this is really what we mean by reinforcement of behavior. You give extra dopamine, you reinforce this choice. If you do the same thing in litamate animals that don't have the right genetic modification for us to tickle dopamine cells, you see no effect. This is just showing it's not an effect of the blue light by itself. And we can use a different opsin, a different virus, to, instead of enhancing dopamine release, to decrease dopamine release, and we show it has the opposite effect. It makes the animal less likely to repeat his choice on the next trial. So that was the effect of dopamine on reinforcement. Confirms that these fluctuations in dopamine really do seem to be being used as reward prediction errors. What about the immediate effect on motivation? So what we find is that if you turn on the laser and you give extra dopamine right at the beginning of the trial, then he is faster to respond to the light, just as if he'd received more rewards recently. Okay. taking. So this is presenting it as hazard rate, how likely he is to initiate a approach behavior if he hasn't already initiated it at each moment in time. And just giving this brief train of extra pulses causes him to transiently be more likely to decide to play this game. Okay, that's what I just said. All right. So the overall conclusions from this talk are as follows. Firstly, that the time-varying fluctuations in dopamine correspond well to a value function, which is estimates of available reward discounted in time. And this value function provides this estimate, and you can use this estimate to influence decisions to work, whether it's worthwhile to the rat to engage in this effortful task or not, whether just do something else. And that the sudden changes in this dopamine value signal are used as reward prediction errors that reinforce choices. Right? And finally, I mean, many people in the dopamine field believe in this strong distinction between phasic and tonic dopamine, that dopamine on different time scales is doing different things, and I no longer believe that. I think that, you know, dopamine, mesolimbic dopamine, in the nucleus accumbens, is dynamically signaling the estimated value of work. That could also be phrased as the opportunity cost. How much are you missing out if you are just slothful and do nothing? And with that, I'll thank my people in my lab again, and thank you very much for your attention. And I hope you could hear what I was saying. But I can't hear you. Hang on a second.
Thank you.
There you go. Thank you.
Yeah. No, I'll open up the stage for any questions anybody might have. So that
please.
Sure. Any questions? So actually, I had one question which I'll just go back. Dr. Berke, if you could turn off the screen.
How do I turn off the screen share?
Yeah, it's done. It's done.
Okay.
No, I was wondering, you have looked at primarily the effect of dopamine in learning and value work. Have you tried, as in how does GABA and inhibitory signals come to play?
So GABA has a huge role at many places within the basal ganglia circuitry. So, for example, at the level of the midbrain dopamine cells, their activity is sculpted by local GABAergic neurons. That's one way. Another way is in the striatum itself. The striatum is fundamentally a GABAergic structure. Almost all the cells are GABAergic, and the local interactions between those cells seem to be critical for shaping which actions are selected. And so GABA, I think the way that I would think about it is that there are certain signals in the brain that are more like scalar broadcast signals, and then there are other signals, transmitters in the brain that are used for the finer details of computations. And GABA is really used more in the finer details of computations, which sets of, which assemblies of neurons within the striatum are able to have a successful bid for behavioral control. I'm not sure if that fully gets at your question, but it's a different kind of role. It's really think of different between vector signals and scalar signals, perhaps.
So it kind of adds on to the rougher dopamine signal, is it?
Well, so find the right way to phrase it. So first of all, GABA is used in all kinds of computations throughout the basal ganglia. In terms of the sort of value and prediction error signals that we're talking about, GABA, several groups have shown that GABA is critical in shaping these the activity patterns of dopamine cells. But we also know that signals within the striatum itself are important in controlling dopamine release at the level of dopamine terminals. So GABA, to some extent, certainly acetylcholine, certainly glutamate, can shape the degree to which dopamine is released from terminals separate from whether the dopamine cell is firing action potentials. and that local control of dopamine release is likely to play a key role in value coding by dopamine. But we don't know very well.
Let's see if anybody else has any questions. Yeah, anybody with questions need to come forward so that.
I have a question.
Please.
Is this experiment, can we conclude anything about human dopamine levels from this experiment?
I'm sorry, I couldn't hear the question very well. I can't bite you, I'm too far away.
Sir, is it possible to conclude anything about the human dopamine levels under such circumstances from this experiment?
Sure. So, thank you for the question. So, of course, we study rats. I happen to like rats, but I don't study rats because I like rats. I study rats because the fundamental organizational principles of these circuits are broadly shared, with humans and all other, vertebrates actually, and maybe even beyond. So we study rats because they're more experimentally tractable, and we can put lasers in their brains and do things like that. But we assume that the same signals are serving the same functions in humans as well. Now, it's harder to assess that in humans. Some people have tried, and there's some evidence that, so for example, you can do some invasive recordings in people who are having brain operations for other reasons, and dopamine seems to have the same kinds of signals in humans as well. Most of the work in this field that's been done in humans is done using fMRI, functional magnetic resonance imaging, and sort of making the assumption that the signals you see with that technique relate to dopamine, but that's not really established. So we assume that it's of direct relevance to humans, but we don't actually record humans, so I suppose we don't know that for sure.
Sir, I wanted to know, see, your study affects, tells us something about performance, human performance and reward and the connection. So what do you think the effects of this study will be on fields like management, sociology, and economics?
I think the effects of this particular study in isolation will be almost nothing on those fields. I would say that's as it should be. So my own opinion about these matters is that too often people want to grab things from neuroscience and apply them to ideas of human performance or management or what have you, but it's not obvious why you should. I think it reflects a sort of reduced status in academia of research that only looks at human behavior without looking at brain stuff. But if I told you that the same things that I just told you were true, but they were true for serotonin and not for dopamine, how would that profoundly affect your management style? I don't think it should fundamentally. I think we're really interested in trying to describe and divulge the architecture of neural systems. But if you want to understand how people behave, I think you should just watch how people behave.
Look, I'll just pop by with a small question here. So you initially talked about firing rates in dopaminergic neurons in the nucleus accumbens. So I remember reading somewhere that the firing rates don't exactly corroborate with the dopamine release.
That's right. Yeah, so that's sort of a fairly recent set of observations. Is really we're only just beginning to understand how different elements of striatal circuitry can control dopamine release regardless of the firing of action potentials of the dopamine cell. So we're really just at the beginning of understanding that process. I think there's several reasons. One of the big questions that comes up each time I give this talk is, well, how come what you see when you measure dopamine release, it seems to be so different from what Wolfram Schultz and other people record when they actually measure the spiking of dopamine cells in the midbrain. And I think that there are several potential reasons for that. One reason is, as we say, because dopamine release can be controlled separately from those spikes. We're only just beginning to understand those processes. The second reason I think is important is because in the typical monkey experiment in which those recordings were made, the monkey didn't really have to do anything. He's sitting there passively waiting for the juice to be delivered. And I fundamentally think that dopamine is not about just sitting around waiting for things to happen to you. I think dopamine is fundamentally about how energized you are to go and get stuff. And I think that there's a series of recent papers that are coming out to support that idea. So I guess what I'm saying with there is that the nature of the behavioral task makes a big difference, potentially. And another way it makes a difference, finally, is just if you're a monkey just sitting waiting for the juice, and the only way you have to assess whether the juice is coming or not is the elapsed time, then it may be harder to assess that compared to a rat that's actually moving around and is getting a constantly changing set of sensory cues, and he can see he's getting closer and closer and closer to the reward. And I think that's why the dopamine is ramping up in a way that hasn't generally been reported for the dopamine cell.
Yes, sir. Sir, I wanted to ask one question regarding in the slide when you compared all the neurotransmitters and how they were related to reward rate and other functions. For exploitation, it was that either left or right were they going or not, sir. But how did you calculate that for cumulative rewards? What was the criteria for calculating the p-value for cumulative rewards for different neurotransmitters?
Was the question, why did we look at cumulative rewards, or how did we look at cumulative rewards?
How is it?
That's very simple. So these are just regression analyses, and in that case, we're just regressing the time course against just the total number of rewards that have been gathered up to that point within the session. So it's almost too trivial an analysis, and it's also not ideal in the sense that it's a little ambiguous. That could mean several different things. It could mean, how satiated you are, how many rewards you've got to this point. It could be how happy you are. You've gotten 133 rewards at this point, or it could be that you're feeling full of sugar pellets after having consumed 133 of them. So, but we wanted to just be able to distinguish that from the signal that was being conveyed by dopamine.
So then, on the overall level, the dopamine signals doesn't change if you take the entire time scale for the experiment.
I only heard part of the question, but I think it was like, does the overall level of dopamine change? Okay, so there's an interesting question here. Over what time scales is dopamine changing? Is there, for example, slow drifts in dopamine that are happening? So we did not see a consistent slow change in dopamine that correlates with anything in our task. So it's not the case that on average across all the rats, the dopamine just generally increases or decreases across the session. So, if that helps.
Okay, thank you.
Excuse me, sir. Hello. Sir, I had actually two questions. As we know that in Parkinson's disorder, the dopaminergic neurons gets affected. So, your recent discovery on this, the dopamine cell firing, can it give some idea about why, the selective vulnerability of only this dopaminergic neurons and why not other neurons?
Sure. I'm happy to tell you what I think the state of the field is on this. I don't directly research this myself. So, do you remember at the beginning I showed a slide showing that just a relatively small number of cells in the midbrain send these very wide projections out to innervate large areas of the forebrain. And because they have these enormous axonal projections, the metabolic load on dopamine cells is unusually large. So they actually have to use a lot more energy than most neurons in the brain. And it's thought that really that requirement for energy is, if you have some genetic vulnerabilities, is likely to lead to toxicity and death of those cells. And for example, there's been some recent work showing that, well, we know that in Parkinson's disease, the dopamine cells that tend to die first are the ones that project up to dorsal striatum. And that's why Parkinson's disease tends to present first with motor symptoms rather than cognitive or emotional symptoms, right? Because those dopamine cells that are projecting to motor parts of the striatum are dying first. And it's been shown that those cells have an even larger axonal projection and even larger metabolic demands than the ones that project down to ventral striatum. So I think that those recent studies are compatible with the idea that if you have a body-wide slight defect or susceptibility to death with excessive metabolic load, then that's going to cause dopamine cells to die first.
Sir, I had another question, I mean, on Alzheimer's actually. So actually, as we know that we use normally radial arm maze to study, I mean, the behavioral aspects of, I mean, control and between the Alzheimer's kind of mouse or rodents we use. So the thing is, I mean, actually, I wanted to know that what's the difference between, I mean, is this working memory, special memory, and reference memory is affected in Alzheimer's, and to what extent?
Sure. So I think what I understood you saying is you're using a maze task to distinguish between different aspects of memory. I'm familiar with some studies like that. So one of the ways that one of the reasons I'm familiar with those studies is because people have shown that different forms of memory that can be assessed in the same maze rely upon different systems within the brain. So, for example, I think what you referred to as reference memory might be that you have a rat in an 8-arm maze or something like that, and some of the arms are always baited, some of the arms always have food, and some of them never have food. Well, it was shown a while ago by Norman White and Mark Packard and people like that, that remembering that kind of reference memory is critically dependent on parts of the striatum, whereas a more working memory task that requires you to remember which ones you've just been to and not go back there is more dependent on hippocampal function. So this was one of the sets of results and ideas that led to what was called multiple memory systems frameworks, and now has been subsumed to some degree within the reinforcement learning literature as what's sometimes called model-free and model-based decision making. So the idea that you have all kinds of experiences, and some of the things you might want to learn require you to throw away some aspects of your experience, and other things you might want to learn require you to throw away other aspects. So I'll give you one example. I hope I'm not speaking too long. One example would be, for example, I drove to work this morning and I had to park my car, right? And so when I make the decision about where to try to park my car, I have to use not what happened yesterday, but my experience over many days of knowing which parking lot is more likely to have an available space. Okay? But when I go to collect my car this evening, it's not useful to me to know which one is usually the best. What I want to know is on this particular day, where did I park my car? So the idea is that those different forms of memory are useful for different kinds of decisions. And that's why we have these different multiple brain systems that are sometimes working cooperatively and sometimes working in opposition to guide, to direct our behavior. I hope that was at least somewhat relevant to the question you were asking.
Sir, I guess I had another question regarding, I mean, say for example in Alzheimer's mouse, a few days, few months back there was kind of discovery that ultrasound kind of reduced the amyloid plaques in the mice. I mean, those kind of mice only had the amyloid plaques. They were transgenic mice and they had only amyloid plaques. I mean, other features of Alzheimer's like neurofibrillary tangles and all were not present. So my question is that will this kind of mouse serve as an effective model system to a person who has, I mean, Alzheimer's with the properties of like neurofibrillary tangles and these amyloid plaques also? Because they experimented exactly on the mouse with amyloid plaques. So, I mean, will the experiment be that effective?
Well, I think it's an extremely important and fascinating question, but it's not in my field of research at all. So I don't feel like I can give an informed answer on it. So I apologize for that.
Thank you.
So I have a question. Sir, initially you mentioned that different stimulating drugs, they induce secretion of dopamine, and dopamine produces reward, like motivation and reward feelings. But in humans, we see that these drugs also produce different feelings like aggression or sadness. So are these also related to dopamine secretion, or are there other factors in play?
I like that question. The short answer is I don't know. The more speculative answer is that, dopamine serves to drive, to release behaviors and feelings that are encoded by loops through these basal ganglia circuits. So if the particular loop that has experiences extra dopamine release is important in driving movement, then you get extra movements. But if you get dopamine release in circuits that are important for emotional feelings and expression of emotions, then you could potentially get enhanced emotionality for that reason. So I would say that dopamine wasn't directly involved in the specification of which emotions you're going to feel, but it might be more involved in enhancing or amplifying the release of all kinds of things, including both overt movements and also certain thoughts and emotional expression as well. That's kind of a lame answer, so I'm sorry I can't do better than that. There is also, I should say, some general association between drugs that cause dopamine release, like cocaine or amphetamines, and feelings of well-being and indomitability, like you can do anything. So a feeling of powerfulness, and I don't think I have a clear understanding or description of how that works right now.
Thank you.
Yeah. Hi, it's me. I just one last question. I was wondering, how long were your sessions with rats? Individual sessions?
How long? Usually about between 1 and 2 hours.
Because I was wondering if you, when you look at dopamine levels, do you, what would you expect the influence of LTP or something like that to be? Would you?
The effect of what? Sorry, LTP?
Long-term potentiation or something like that.
Sure, that's a good question. So I think one of the things you're getting at is what is the role of, moment-by-moment plasticity on behavior on different timescales. So, the simplest way of thinking about this action of dopamine on both learning and motivation would be that the learning aspect is done via things like LTP. So that especially spike timing dependent plasticity can be modulated by dopamine, and it would make sense as a mechanism of adjusting your expectations of reward to have dopamine serve that function in this task. Conversely, we hypothesize that the immediate energizing and motivating effects of dopamine are by affecting the excitability of neurons rather than directly affecting their plasticity. So at least the way we think about it is that twin functions of dopamine are really operating via plasticity and excitability of synapses and neurons within striatum respectively. So we think LTP is directly important. We know that LTP seems to be involved in the reinforcing actions of dopamine, but we haven't gone much further than that.
Thank you. Any further questions?
Thank you once again for the invitation.
Anyway, I think that's all the questions we have today. Thank you, Dr. Berke, for joining us for this session tonight. And
thank you very much.
It was a fun.
Bye-bye.